Ya, Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera bagi kita semua. Shalom, Om Swastiastu, Namo Buddhaya, salam kebajikan. Selamat pagi Bapak-Bapak, Ibu-Ibu, dan teman-teman semua, dimanapun Anda berada, di seluruh Indonesia maupun di luar negeri. Saya Eni Buhari, dan saya akan menjadi moderator di acara ini. Alhamdulillah kita semua bisa berkumpul bersama secara virtual. Semoga kita semua tetap sehat dan tetap produktif di masa pandemi ini. Sebagai bagian dari kegiatan Supplemental Technical Assistance untuk USAIDC Project, Kementerian Kelautan dan Perikanan Republik Indonesia bekerja sama dengan the University of Hawaii yang berjudul Histology-Based Rapid Reproductive Analysis for Tropical Fish. Tujuan dari pelatihan ini adalah sebagai bagian dari pengembangan kapasitas dan sharing pengetahuan di bidang ilmiah, khususnya untuk para peneliti, dosen, mahasiswa, teknisi dan analis laboratorium maupun pengelola atau manajer perikanan dan kelautan, serta juga para praktisi lapangan di bidang kelautan dan perikanan yang sehari-hari bekerja dalam kapasitas masing-masing di bidang aspek reproduksi ikan dan biologi ikan dan juga pengkajian stok ikan. Pelatihan ini terdiri dari 2 modul yang akan diberikan dalam 4 hari. Modul pertama diberikan dalam 2 hari, 18 Januari dan 21 Januari. Dan modul kedua akan diberikan pada tanggal 1 dan 4 Februari 2021. Masing-masing 3 jam per hari, dari jam 9 pagi sampai jam 12 siang waktu Indonesia bagian Barat. Materi akan diberikan langsung oleh tim instruktur dari the University of Hawaii dan bisa diakses langsung di portal Lau 5 yang saya yakin Bapak Ibu sudah menerima emailnya. Sebelum kita lanjut, kami ingin jelaskan sedikit aturannya atau house rule di pelatihan ini. Yang pertama, mohon agar para peserta mengklik speaker view di pojok kanan atas dari layar Zoom Anda agar bisa melihat dengan jelas layar dari orang yang berbicara. Sehingga nanti Bapak Ibu bisa langsung melihat materi training yang akan disampaikan oleh tim. Yang kedua, semua mic peserta dibisukan atau di-mute secara otomatis selama acara, sehingga tidak masuk suara-suara latar belakang. Namun nanti di sesi pertanyaan, peserta bisa mengangkat tangan dengan cara mengklik tombol angkat tangan di bagian bawah layar Zoom ya, raise hand, dan kemudian nanti moderator akan memilih secara random beberapa orang untuk bisa mengajukan pertanyaan secara live dan mic Anda akan dibuka sampai pertanyaannya selesai. Kemudian jika tidak ada kesempatan bertanya secara live, Bapak Ibu juga bisa mengetik di dalam kolom Q&A di bagian bawah layar Zoom Anda ada Q&A, bisa langsung mengetik pertanyaannya di situ, ditujukan kepada siapa, dan bisa dalam bahasa Indonesia juga, nanti dari tim kami akan menerjemahkan ke bahasa Inggris. Pertanyaan lebih lanjut jika masih banyak lagi pertanyaan, bisa juga dikirim melalui email ke alamat email tanya.histology@gmail.com. Dan yang terakhir, training ini diberikan dalam bahasa Inggris. Namun, jika Bapak Ibu melihat di bagian bawah panel Zoom sebelah kanan ada ikon logo globe, di situ ada pilihan bahasa, baik bahasa Indonesia, bahasa Inggris, dan Bapak Ibu bisa memilih bahasa yang dikehendaki. Nah, tanpa berpanjang-panjang lagi, saya akan memperkenalkan Bapak Profesor Insinyur Syarif Wijaya, PhD, Kepala Badan Riset dan Sumber Daya Manusia Kelautan dan Perikanan KKP yang akan membuka acara pelatihan ini. Profesor Syarif Wijaya sudah menjadi Kepala Badan Riset dan Sumber Daya Manusia Kelautan dan Perikanan sejak tahun 2018 hingga sekarang. Sebelumnya, beliau juga menduduki jabatan penting sebagai Direktur Jenderal Perikanan Tangkap di KKP. Beliau meraih gelar PhD-nya di tahun 1992 dari the University of Strathclyde di Glasgow, UK, dalam bidang Computer Aided Ship Production. Langsung saja, waktu dan tempat kami persilahkan. Profesor Syarif Hidayat.
Baik, terima kasih Ibu Erni. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi, salam sejahtera untuk kita semuanya. Om Swastiastu, Namo Buddhaya, salam kebajikan. Very good morning to all of you, the distinguished speakers, participants, ladies and gentlemen. Yang saya hormati Dr. Alan White, Director for the USAID project, the head of the Fisheries Research Center, Ibu Hiter Agnes from the Walton Family Foundation, Dr. Gina Green, Tetra Tech ARD, Associates in Rural Development, Dr. Eric Franklin from University of Hawaii, Dr. Ken Lochner from Bishop Museum, Hawaii, Dr. Ross Langston from Windward Community College, Hawaii. Thank you for coming. Dr. Ken and Dr. Ross, also Dr. Eric, thank you. Ibu Erni Bukhari, USAID Project, all participants, ladies and gentlemen. Pertama kali, marilah kita panjatkan puji syukur kehadirat Allah Subhanahu Wa Taala, Tuhan Yang Maha Esa, atas segala nikmat dan karunia-Nya, sehingga kita bisa bertemu pada pagi hari ini. All the blessed from the Almighty God for all the healthiness and happiness and protection us from the pandemic COVID-19. We are still working and more productive for the shape of the peoples of all over the world. And today we will discuss about the analysis reproductive, histology-based rapid reproductive analysis for tropical fisheries. Bapak Ibu sekalian saya hormati, sebagai negara kepulauan, Indonesia diberikan anugerah kekayaan laut yang berlimpah, sumber daya ikan yang sangat beragam dan produktif, menjadi landasan bagi ekosistem laut, dan yang lebih penting lagi, kegiatan penangkapan ikan merupakan mata pencaharian pokok bagi lebih dari 3 juta nelayan di negara kita ini. Hal ini memberikan sumber pendapatan bagi nelayan, menjaga kestabilan pangan nasional, dan sumber protein yang luar biasa bagi sekitar 267 juta penduduk Indonesia, sehingga kita dapat mencapai keamanan dan kedaulatan pangan secara nasional, serta memberi kontribusi kepada komunitas global. So I would like to mentioning in this paragraph that Indonesia is an archipelagic state. We have very rich and biodiversity in the marine and ocean resources. Now as a resource for the food security for the people of Indonesia and also for the people of the world. So tentu sebagai lembaga atau kementerian yang menjaga laut, KKP akan memastikan keberlanjutan dari ekosistem kelautan ini bisa berjalan dan tidak rusak, serta perikanan kita tetap produktif. Untuk memastikan perikanan kita dikelola dengan baik, diperlukan kebijakan yang dapat secara jelas menjabarkan fungsi-fungsi dari pengelolaan perikanan berdasarkan saran ilmiah dari berbagai tataran, baik di tingkat kabupaten, tingkat provinsi, tingkat BPP, wilayah pengelolaan perikanan, tingkat nasional, bahkan pada tataran regional sebagaimana halnya dilakukan oleh Regional Fisheries Management Organization. So all the resources from the sea, we have to manage properly to make sure that sustainability of the capture fisheries can be implemented in all aspects for the peoples and communities. And we are following the rules and regulation, even in the national level and also regional level, like mentioning in Regional Fisheries Management Organization, RFMO. Untuk terus mendukung kebijakan pengelolaan perikanan ini diperlukan time series data yang memadai, berkualitas dan dapat dianalisis bagi keperluan pengelolaan perikanan. Metode ilmiah dan analisis yang kuat seyogianya dilakukan dengan proses yang transparan dan inklusif guna menghasilkan recommendation dan opsi-opsi pengelolaan perikanan yang terbaik. So we are rely on the data, time series data, and scientific analysis will be useful to give the proper recommendation for the management of the fisheries in Indonesia. Especially the data should be prepared in transparent and inclusive way, inclusive manner for all the peoples. Salah satu metode ilmiah dan analisis yang kuat ini adalah analisis reproduktif cepat berbasis histologi untuk ikan-ikan tropis yang mampu mendukung proses pengkajian stok ikan dalam rangka menyusun strategi pemanfaatan sumber daya perikanan. Pengkajian stok ikan yang kuat dan mumpuni sangat dibutuhkan untuk dapat memastikan bahwa laut Indonesia terus produktif secara berkelanjutan dan pada gilirannya akan dapat mengamankan masa depan sektor perikanan, meningkatkan ekspor, memastikan keamanan dan kedaulatan pangan, serta melindungi pekerjaan pada sektor perikanan secara terus-menerus bagi anak cucu kita. BRSDM merupakan institusi terdepan yang mengawal dan memastikan proses ilmiah untuk pengelolaan perikanan tangkap dilakukan dan dijadikan sebagai pijakan bagi pengelolaan perikanan secara nasional. So we prepare some recommendation based on reproductive analysis, rapid reproductive analysis based on histology for tropical fisheries, which can be supported for the stock assessment analysis. So by using this technique, we are hoping that the management of fisheries of Indonesia can be done with a sustainable manner for the future of the next generation. Saudara-saudara sekalian yang saya hormati, USC Project merupakan proyek 5 tahun 2016-2021 antara pemerintah Amerika Serikat dan pemerintah Indonesia dan bertujuan untuk mendukung pemerintah Indonesia dalam memperkuat tata kelola sumber daya perikanan dan kelautan serta konservasi keanekaragaman hayati di wilayah pengelolaan perikanan WPP 715 Indonesia dengan didukung oleh Walton Family Foundation, sebuah inisiatif teknis tambahan atau supplemental technical assistance terhadap USC Project diarahkan untuk menambah capaian-capaian C Project dalam bidang ilmiah dan inovasi dan teknologi. So I would like to share this information that we are now having a USC Project 2016-2021 between United States of America, USA, and Indonesia, and we are supported by the Walton Family Foundation. We are having some pilot project in the WPP 715, yeah, Fisheries Management Zone 715 is Tomini Strait, ya, Teluk Tomini, yang as a pilot project for the Indonesian archipelagic. Memasuki akhir proyek dalam rangka berbagi pengetahuan dan pengalaman, supplemental technical assistance terhadap USC Project bersama dengan Kementerian Kelautan dan Perikanan serta didukung oleh the University of Hawaii akan menyelenggarakan pelatihan secara daring. Analisis reproduktif cepat berbasis histologi untuk ikan-ikan tropis yang akan diselenggarakan dalam 4 sesi mulai tanggal 18 Januari hingga 4 Februari. Thank you to the University of Hawaii to support all the efforts. Now today we will have a 4-day session for the seminar to share and to disseminate the result of the studies, yeah, from the Fisheries Management Zone 715 Indonesia under the C Project and Walton Family. Finally, I would like to thank to all the distinguished speakers, all the tutors, and all participants for having to share the information concerning about the result of this study. To the people of Indonesia, thank you very much, and I really hope that the seminar will be fruitful discussion and fruitful results for the people of Indonesia. Melalui kesempatan ini, dengan mengucap Bismillahirrahmanirrahim, dengan ini saya resmi membuka acara pelatihan secara daring. Analisis reproduktif cepat berbasis histologi untuk ikan-ikan tropis. Terima kasih. Wassalamualaikum warahmatullahi wabarakatuh. So officially open, yeah. Thank you, Dr. Ken, Dr. Eric, and Dr. Ross Lapson. Thank you for your participation. I really hope not only by Zoom. Next time we can meet, yeah, after the pandemic, and we can fly to Hawaii and discuss about the development of the capture fisheries for the future of Indonesia and US. Thank you very much. Terima kasih Bu Erni, saya kembalikan kepada Ibu.
Terima kasih Pak Syarif Wijaya atas sambutannya dan pembukaannya. Thank you very much. Berikutnya saya akan memperkenalkan tim instruktur yang akan memandu acara pelatihan selama 4 sesi ini. Pertama, saya akan memperkenalkan Dr. Eric Franklin atau yang biasa kami sebut dengan nama Pak Eric. Pak Eric adalah seorang Associate Research Professor di University of Hawaii yang karyanya berfokus pada identifikasi solusi pengelolaan sumber daya berkelanjutan untuk ekosistem laut tropis dengan menggunakan pendekatan ekologi, ilmu perikanan, dan geospasial. Sejak 2013, Dr. Franklin bertugas di Komisi Ilmiah dan Statistik dari Dewan Manajemen Perikanan Regional Pasifik Barat Amerika Serikat dan memimpin review pengkajian stok ikan untuk perikanan terumbu karang dan ikan laut dalam di Hawaii serta di wilayah Pasifik Amerika Serikat. Sejak tahun 2019 pun, Pak Eric juga telah bekerja sama secara langsung dengan teman-teman kolaborator Indonesia, khususnya teman-teman anggota Komisi Nasional Pengkajian Sumber Daya Ikan atau Komnas Kajiskan, untuk mengidentifikasi perbaikan proses review pengkajian stok ikan untuk Indonesia. Selamat datang Pak Eric di Virtual Indonesia. Selanjutnya yang kedua adalah Dr. Ken Longenecker. Dr. Longenecker atau Pak Ken adalah seorang Associate Researcher di Bishop Museum di Hawaii dengan bidang kepakaran informasi biologis untuk mendukung konservasi dan pemanfaatan sumber daya biota laut yang berkelanjutan, khususnya untuk ikan-ikan karang. Beliau memiliki pengalaman lebih dari 20 tahun untuk bidang riset analisis reproduktif berbasis histologi, analisis population size structure, analisis makanan ikan dan taksonomi ikan dan krustasea. Pak Ken juga banyak melakukan riset dan pelatihan berbasis histologi di berbagai negara, diantaranya di Samoa, Mikronesia, Fiji, Guam, Hawaii, dan Papua Nugini. Pak Ken meraih gelar PhD-nya di bidang zoologi di tahun 2001 dari The University of Hawaii. Dan di pelatihan ini, Pak Ken akan memberikan penjelasan tentang bagaimana cara memproses spesimen ikan untuk analisis histologi dan juga cara melakukan analisis statistik dengan menggunakan Excel. Dan yang terakhir adalah Dr. Ross Langton atau Pak Ross adalah seorang profesor biologi di Windward Community College di Hawaii. Beliau juga memiliki pengalaman 20 tahun di bidang life history dan biologi reproduksi ikan-ikan karang. Bersama-sama dengan Pak Ken, Pak Eric, Pak Ross juga ikut memimpin pelatihan dan lokakarya tentang analisis berbasis histologi untuk jaringan gonad ikan, untuk mahasiswa universitas dan juga untuk manajer perikanan. Beliau meraih PhD-nya di bidang zoologi dari University of Hawaii di tahun 2004, dan di pelatihan ini Pak Ross akan memberikan materi pelatihan tentang cara memproses histologi gonad dan analisisnya. Langsung saja saya berikan ke Pak Eric untuk memimpin pelatihan ini. Pak Eric, time is yours.
Terima kasih, Bu Eni, and thank you, Pak Syarif, for your wonderful opening statement. My colleagues and I are honored to be invited to present this training to our friends in Indonesia. My name is Eric Franklin. I am an associate research professor at the University of Hawaii and serve as the technical lead for fisheries science for the supplemental technical assistance to the USAID project managed by Tetra Tech, funded by the Walton Family Foundation. This training is part of the Supplemental Technical Assistance Project. The training will provide you with an introduction to the histology-based analysis of tropical fishes that can provide life history information that is useful for marine conservation and fisheries management. These methods have been developed and refined over the last 10 years in the Indo-Pacific region by the instructors. The approaches are described in detail in a standard operating procedure document that we will share with you. The training is presented in 2 modules. The first module will provide an overview of the anatomy, development, and classification of fish reproductive structures. We will present microphotographs of fish gonad sections during instruction and provide interactive feedback to assist participants during the training. The second module will give an introduction to the statistical analysis of reproductive data, including methods to calculate size at maturity, sex ratios, fecundity, and other traits. We will provide an example data set and use it to demonstrate the different analysis methods. For both modules, an exam will be offered for participants to test their knowledge of the course materials. The exams will be offered online for the rest of the year and can be taken as many times as necessary to achieve a passing grade. Once the exam is passed, the participant will receive a certificate of completion issued from the University of Hawaii. Thank you for participating in this course. We truly look forward to sharing the materials with you and helping you to learn more about tropical fish reproduction. So it's time to begin. Thank you.
Pak Ken, now it's your time, Pak Ken.
Thank you, Annie, and Salamat Pagi. Thank you for joining us here today, and I'm very pleased to be able to speak to everybody. What I'd like to do right now is to give a very brief overview of the histological techniques that we use. There's 2 reasons that I want to give this overview. One is because we are going to spend today and the next time we meet looking at photomicrographs or pictures of microscope slides with sections of fish gonads on them. And I'd like to be sure that everybody understands how we produce those slides or how we produce those photomicrographs. In other words, what is this process of histological analysis? The other reason is because we're going to spend the 3rd and 4th sessions reviewing statistical analysis. And I would like to be sure that everybody understands how we gather the data and how we manage the data so that we can successfully perform those statistical analyses. So with that, we're going to talk about specimen processing, or the big picture is how do we get from a bunch of fish specimens to the point where we have those slides of fish gonads that we're going to be analyzing in the first session. And then how do we take care of the data that we gather along the way? I'd first like to start with a basic question: what is histology? And the answer is simply that it's the study of microscopic anatomy of fish tissues. In other words, these are the slides that we are producing with histology-based reproductive analysis. And it's the analysis of these slides that are going to give us much of the data that we use and will drive much of the analyses that we do in the second module. So I'd also like to answer the question, why histology? Some of you may be familiar that you can go out and do reproductive analysis by simply cutting open a fish and looking at its gonads, such as what we have right here. The reason is that we've found when doing our own analyses, we will look at the gonads in this macroscopic state or just with visual analysis, and then we will compare our results to the exact same specimen when we've done a histological analysis, and found out that we are wrong 40 to 50% of the time for every species that we've ever examined. And at this point, you might think, well, maybe Eric, Ken, and Ross just aren't that good at reproductive analysis. There's other people who've done similar analyses and find the same degree of inaccuracies. So to get very good information or very good data necessary for stock assessments, it's necessary to take this histological or microscopic approach. Another reason why histology is necessary is because in fact, I would say many species of reef fish at least, change sex as a natural part of their lifespan. They'll start off as a female and at some point change to a male, or the other way around. And you have to do histological analysis in order to confirm that this sex change takes place. Here's a brief overview of what the whole process looks like with our histology-based analysis. I'm going to go into a lot more detail in a little bit in the next couple of slides. And then don't worry if you're not quite grasping everything, because as Eric says, we have produced a standard operating procedure. It's going to have text description of what we do in quite a bit more detail than I'm going to speak about today. And it will also have many images that will be helpful to you as you're trying to learn this process of identifying fish gonads. In fact, it may be helpful when you do the exam. So we start with a bunch of specimens. Here we are. Oh, I also wanted to mention that Ibu Eni told you some of the places where we've worked in the past, and what you're going to see are images from work that we've done throughout the Pacific. So here we are in Papua New Guinea, working on a small species of snapper, processing those specimens, getting lengths and weights, removing their gonads, and then we put the gonads into tissue culture plates. And the first thing we do is fix them, then we dehydrate them, then we go through a process called infiltration, and finally we embed them in plastic. And here's where our approach differs from some of the other approaches that you might be familiar with if you know about histology. It's very common to use what is called wax embedding. Wax embedding takes a lot of equipment that would be very difficult to operate in remote locations. So we use what is called plastic embedding. And here's what the tissue sections look like after they've been embedded in plastic. We get what we call stubs. We put these stubs into a special machine called a microtome. And that microtome enables us to make very thin sections of the gonads. Then we mount those gonads onto a microscope slide, we stain them, and we come up with images such as these that are going to be what you're going to learn how to analyze today. So let's look in a bit more detail about each of those steps and also how we manage our data. And I just wanted to show you an example right now of what the data for a single species would look like. The sorts of data we collect. Don't get overwhelmed by this right now because I'll be zooming in on each of these columns as we discuss them through the remainder of my presentation. The first thing that we do when we have a new specimen is assign it a code and a specimen number. Our code is the two-letter country abbreviation for the place where we're working or where the fish was collected, and then a two-letter species abbreviation. And then we add to that just a number. The numbers go in sequence just in the order in which we process the specimens. So here's an example. We were working in Fiji. Its two-letter country code is FJ, and we were working on Latjanus gibbus. So we made an abbreviation of the first letter of the genus and species name, LG. This happened to be the 134th specimen that we looked at, so we make this little specimen tag, just FJLG134. And this specimen tag is crucial. This tag follows the tissues from the fish the whole way through the process until we do the sectioning on the slide, and then that number is written down the slides. So if you don't keep track, or if you lose the specimen number, you lose all the data that were collected along the way, which is a tragedy when you've put so much work into it. This is just an example. I'll get my head out of the way just a second here. This is just an example of what a species code would look like. And here we've broken it down into different columns, a column for code and a column for the specimen number. So this is a made up data set for the purposes of training today. We're saying that we're working with the species Latjanus langstonii and that we're working on the species in Indonesia. So the two-letter country code is ID, then we would add LL for Latjanus langstonii, and you can see as you go down the specimen column that we just sequentially increase the number each time. The next thing that we record is the date, and that's different columns for the day, the month, and the year of collection. So here is one of the species that we're planning on working on in Indonesia. we ever get to the point where we can travel again. This is Epinephelus fasciatus. And here you can see that and they're frozen until we arrive. You can see that the collector wrote down the date of collection, and they're going to be sitting there in the freezer waiting for us to process them when we finally do get to make it to Indonesia, but the date's going to be with all those specimens. And here we just have separate columns for the day, the month, the year. And we use these data for trying to determine spawning periodicity or peaks in spawning. You might look at monthly variation. If you had a multi-year project, you could look at annual variation in spawning. Or here we use the day because many times we see that spawning is associated with the moon phase, and we can use the day and month to calculate the moon phase and then look at how spawning might change with moon phases. Now we're actually ready to start collecting some data about the fish. The first thing we do is measure its length, and that's in tenths of a centimeter, and we put that in a column called length. And a lot of what we're doing with the histological analysis, a lot of our goals in this whole process is to find out how reproduction changes with length. What is the size at maturity? How many eggs does a female produce with a given length? Things like that. Another bit of data that we collect is weight. Here we are working in American Samoa. Here are some of the trainees that we had in American Samoa. We simply weigh the fish to the nearest gram on a hanging spring scale and enter that in the weight column. Now we're going to start dissecting the fish. We're going to take the gonads and remove them and weigh the whole gonad on what we call a jeweler's scale, and we weigh that to the nearest thousandth of a gram. And here we enter the whole gonad weight in this column. And you're going to see a lot of these data later on when we do the data analysis or the statistical analysis part. While we have the gonad on the scale, we also do a macroscopic evaluation of its sex and its maturity status. Now, you might remember that I spent quite a bit of time, or a little bit of time at the beginning, saying it's very important that we do this analysis on a microscopic level or do histological analysis. Why would we bother with the macroscopic analysis? Well, there's two reasons. One is because that's the thing that enabled us to compare our results when we did macroscopic versus histological analysis. The other reason is that if it looks like we have a mature female, then we take a special subsample of her gonads to do the fecundity analysis or to try to predict how many eggs a female produces each time she spawns at a given length. So here we have a column called macro sex, which stands for macroscopic sex determination or evaluation. And we have four categories. They can begin with either a capital F or a capital M, which stands for female or male. And then there's lowercase MAT or IMM, which stands for mature or immature. So here we have a mature female, mature male, immature male, immature female was how we evaluated that macroscopically. So I just mentioned that if we think we have a mature female, we take a special sample of her gonad for the fecundity or egg count analysis. And this is what and we only do that for what we think are mature females. Okay. So if we think we have a mature female, we take about a 1 centimeter thick cross section through her ovary. We weigh that just like we did the whole gonad sample to the nearest thousandth of a gram. And then we put that gonad sample into a storage container. And either if we work at a place with no refrigeration, begin processing that right away. If we work at a place where we have a refrigeration, then we would just stick it in the freezer until we do the histological analysis. And right here, you'll notice there's another tag that's been created, and that goes into the into the container along with that gonad subsample. So again, it's very important that those specimen numbers accompany your samples the whole way through during the processing. And we just record the sample gonad weight down here. So you'll see that every time we have a sample gonad weight, it's across from what we thought was a mature female. Our next step is to histologically determine the sex and the maturity status of that specimen. Now we're going to take a much smaller sample of the gonad, and it's about 3 millimeters on a side, a cube about 3 millimeters on each side. We put that sample into the tissue culture well plates, and you can see if you look closely here that the specimen tag is now in the well plate with each one of these specimens. We add a fixative. We allow that fixative to fix the tissues for at least 24 hours. Then we go through what is called a dehydration process. We remove the fixative and add alcohol in increasing concentrations until we're up to 95% alcohol. After that, we remove the alcohol and we go through a process called infiltration. And now we're starting to work with some of the plastic compounds. We work with the plastic resin and put it into the tissue well culture plates. Do that for a couple of changes. And when infiltration, that's about a total of 24 hours as well. When that infiltration is finished, we then take the specimens and transfer them over to another type of container called a beam capsule. So the specimen plus its specimen tag goes into the beam capsule. And then we add catalyzed resin. So it's in a liquid form. After an hour, maybe a little bit more, that turns into a hardened plastic, which you can see right here is one of the stubs. This is the hardened plastic. The tissue sample is inside of that, along with the specimen tag. We put that stub into the machine called the microtome, and this is the chuck holding the stub, and down here is what we call a glass knife. This is a specially produced piece of glass called histology grade glass. We break that in a very specific way so that we have a very sharp edge across this top surface. And the machine just takes the stub down across the knife and again and again, and then comes increasingly closer to that knife. So the sections you just lift off the knife, place onto a slide, like here. We stain those slides, and then when the staining process is finished, we have the slide that we're going to look at. And this again is a picture or a photo micrograph of one of those sections, or actually in this case, probably just a small portion of one of those sections as it will be seen under the microscope. And that's what we're going to learn how to evaluate today. So here you can see we have another column called histosex, and this is now the histologically determined sex and maturity status. We use the exact same codes we used over in the macrosex column. You can see that in most cases we agreed, but every now and then there's a little bit of disagreement. Here we thought we had an immature female, but it turned out to be a mature male. Okay. Here we thought we had an immature male, but it turned out to be a mature male, things like that. The other thing we do, and this is only for the females, is evaluate the egg stage. And Ross is going to show you in a lot more detail how to evaluate the egg stage. But when we do, the reason we record this is that when we do fecundity analysis, we only do estimates, and that was the egg count analysis. We only do estimates for females that had stage 4 oocytes present or stage 4 eggs present. So here you can see that, oh, we didn't have one there, sorry. There must have been a mistake. So here we had, we estimated that it was a mature female. So we did all the gonad sampling, found out that it was a 4A female, that she had stage 4 eggs. So we went ahead and did some fecundity analysis then. There's some other places where you'll see that basically we thought or maybe there's not any examples here. I apologize, I thought I had one. There are other places where we did the analysis, found out that it wasn't a stage 4A female, so we didn't include her data in the fecundity analysis. So when I talk about the fecundity analysis, what I'm really talking about is batch fecundity. And that is, we're trying to estimate the number of eggs that a female would release on the next occasion that she spawns. So what we did was took that gonad subsample that was already weighed, that was the 1 centimeter thick cross-section through one lobe of the ovary. We add an enzymatic solution that digests the connective tissue from that ovary and only leaves behind free oocytes or free developing eggs. When that digestion process is finished, we pour this solution into a different container, dilute it, and then use this instrument called a Stempel pipette to take samples from that solution, from that diluted solution. And these are designed such that when you release the plunger, it will take exactly 1 milliliter of solution from that diluted container. What we do then is put that 1 milliliter solution under a microscope and count the number of the largest eggs that we can see. And we do that 10 times so that we get what we hope is a representative sample of the egg count from that dilution. And here's what the data would look like. You'll notice that this is in 2 colors. Yellow, like I've been showing you so far, is the data that we entered. And over here are some calculations that the Excel spreadsheet automatically does for us. So we enter dilution, and we found that when we dilute that 1 centimeter cross section using 400 milliliter dilution, works out very well for us. And then here's the egg counts from the 1 milliliter subsample that we took from there, done 10 times. This first green column is simply the mean of all of these counts. Okay. And then the last column uses several of the cells in this row to estimate batch fecundity, or the total number of eggs that that female would produce in her next spawning event. And I'd like to explain what that is right now. I'm in the way again, so I'm going to make myself go away. And the formula that we use is that fecundity equals the mean number of oocytes, or developing eggs, per milliliter of that sample, the dilution volume that we used, multiplied by this quantity of the whole gonad weight divided by the sample gonad weight. So this estimate here is based on this number, the mean number of oocytes per milliliter, times the dilution volume. And that should give us an estimate of the total number of oocytes that were in that diluted volume of liquid, or the total number of oocytes that were in that 1 centimeter cross-section through the single lobe of the ovary. What we need to do then is multiply up and estimate how many oocytes were in the whole ovary. And we do that by multiplying by the whole gonad weight divided by that sample gonad weight. And that gives us the final estimate. The final thing that we do is add some notes if there was anything noteworthy to add. And that would be in this last column. Here it turned out that when we did the histological analysis, it turned out that the tissue that was sampled wasn't gonad. So over here, you can see that we've removed the whole gonad weight, and that we've changed the sex and maturity status to UNK, which stands for unknown. We couldn't determine it because we weren't looking at gonad tissue. Another way that some of that error is introduced into that microscopic analysis. Here we were pretty sure we knew what we had, and it turns out that we weren't even looking at gonad. Let's see. And here's another example that I wanted to show you. I mentioned a little bit earlier. Oh, I apologize. I'm going to go on. So that, I believe, is the last of what I would like to say. So with that, I'm going to ask if there are any questions, and either I or my colleagues, Ross and Eric, would be happy to answer them for you. And thank you very much for your attention, and I hope you enjoy the rest of the program.
Thank you very much, Ken. So right now we have several people raising their hands. So I will randomly choose 3 people to ask the question, starting with Petronella M. Paja. Please, Ibu Petronella, mohon dibuka mic-nya. Petronella Paja. Oke, kalau gitu saya skip dulu ke Rumondang Tampu Bolon. Mohon dibuka mikrofonnya. Rumondang Tampu Bolon.
Iya, Bu.
Ya, silakan tanya, Bu. Tadi Ibu angkat tangan, jadi saya pilih. Saya akan memilih secara random 3 orang.
Udah jelas sepertinya, Bu.
Tadi Ibu mengangkat tangan, ini sekarang udah sesi pertanyaan. Jadi saya akan memilih 3 orang secara random untuk bertanya. Silahkan sekarang Ibu bertanya karena saya memilih Ibu. Bisa mengajukan pertanyaannya, Bu?
Pas, Bu, pas.
Pas, oke. Berikutnya, Merdeka Agus Saputra. Silakan bertanya, Pak Merdeka.
Yes, thank you so much for the opportunity, Dr. Eric. There is one question that I want to ask. First is, I think in Indonesia, we don't have this kind of sophisticated technologies yet, because based on my experience, we are still using conventional histological method to look at the gonad fish maturity. And there is one interesting point that you explained about the gonad sample preparations, and there is a, how do you say, after you prepare it with the fixative substance and then alcohol 90%, and it becomes kind of like a plastic form of the gonad. What I want to ask is actually how long does this well-prepared gonad tissue last? And then can we still use this microscopic gonad tissue for further observations? Yeah, so that's my questions here. Thank you so much.
We are not, and thank you for the question. I don't know that we know how long the embedded tissue will last, and I'm going to ask Ross if he has any input on that. As far as the slide tissue that we make, we would then use a mounting medium and put a cover slip on top of that, and that should last indefinitely, the actual slides that were produced. And just to be sure that I didn't misrepresent what I was trying to present, it's not the whole gonad that has been plasticized. It was just that small 3 millimeter cubed section that was plasticized. So if you had some remaining gonad tissue that you would be interested in looking at later on, I would suggest that you just fix whatever was left of the gonad that wasn't placed through this process, and then you could go back and look at that later on. Probably fixative and then placement and storage or preserving in alcohol. And with that, I'll turn it over to Ross to see if he has any thoughts on how long those stubs would last.
Yes, can you hear me, Ken? Okay, so I think the stubs, as long as they're kept on desiccant, we've gone back and resectioned a couple years later. So as long as you don't leave them out to absorb water, and they are hydroscopic, I think it should be fine that you can just hold on to those stubs indefinitely. They will get harder as they dry out, but you can always just humidify them and then section them. So I think they're good indefinitely.
Okay, thank you for your answer, Ross. Next person is Gusasta Arnenda, please. Gusasta Arnenda.
Thank you very much for your... Hello, can you hear me?
Yes, please, go ahead.
Yes, thanks for your opportunity. I want to ask about how to select gonad for the fecundity, because in LRPT or in my research, we use different classification to fecundity. So if you saying that fecundity is the spawning gonad, but we have another classification about the spawning gonad. So what is the be used to fecundity gonad first to the sample, and then is about the how to select the gonad.
So I don't know if this came across during the presentation, or maybe it didn't look that way from the data that I showed you. When we are going through and for any reason think that we have a mature female, we take that cross section, that small subsample of the gonad for potential fecundity analysis. And then it's especially handy if you're working in a place where you can freeze that. We go through, do the histological analysis first, and find that if we have those mature oocytes, the ones that are ready to be spawned, then those are the only ones that we actually use to digest with the enzymatic solution, and those are the only ones that would be counted. So if I were to show you that all of the data that we collected during the whole process, basically anytime you saw FMAT in our gross macrosex column, there would have been a sample gonad weight associated with that. What we did then was, because we weren't doing fecundity analysis on that specimen, we deleted it from the data set. Did that answer your question, or do you have a follow-up question to that?
It's just one question, Ken. So, nanti kalau misalnya mau ada pertanyaan lebih lanjut, Pak, bisa dikirim ke Q&A atau lewat email ya. Now, last person, I would like to call Wulan Kuago. Wulan Kuago, please ask your question.
Hi, hello.
Yes, go ahead.
Thank you very much for the presentation, such a great information. And I think I can see that we have so many questions here in Q&A box. And I do also have lots of questions regarding the procedure. So I think my question is just straight away to the SOP. You mentioned about SOP when you started the slides, the presentation. So will it be shared to us, the detailed procedures or SOPs here? Because I think if we could have those detailed procedures or SOPs, then it will be very useful for us. And I think it will also answer the, those questions in Q&A as well. So yeah, thank you.
Thank you for that question, because it was a very good one. And if you've been able to log into Laulima already, which is the site that we're using for the exam, there are also resources posted on that site. And the SOP is already, the English version of the SOP is already posted on that site. And within days, we should have a Bahasa Indonesia version of the SOP as well. So that will be available to you, and we hope that you download it, and we hope very much that you use it.
Brilliant. Thank you very much.
Terima kasih Ibu Wulan. So I think now we have reached to the end of the first part of the training. We now have a 5-minute bio break, and we'll come back in 5 minutes. Thank you. Selamat siang Bapak Ibu semua. Kita sekarang akan lanjut ke sesi berikutnya. Dr. Rose Langton yang akan memberikan presentasi training berjudul Identifying Female Gonads. Please, Ross, time is yours.
Okay, can everybody hear me?
Yep.
Okay, so at this point in the presentation, we're going to talk about the steps needed to take that slide of gonadal tissue and classify it as either gonad or not gonad, ovary or testis, and then be able to say whether it's mature or immature. So at this stage, you need a microscope, and ideally it should be a microscope with a 4x, a 10x, and a 40x lens. You really need the 40x lens in order to be able to identify spermatozoa, which is part of the thing about identifying mature males and distinguish them from immature males. You also need your microscope slides, which we have here, with your stained tissue sections on there. Now, if you're familiar with regular wax histology, you've probably used hematoxylin and eosin. That requires several steps with the resin-embedded specimens that we use, so we usually just use one dye called toluidine blue. In either case, we end up with one slide per specimen and about 10 sections on there spaced out evenly through the gonads. Ken talked about earlier, we have the specimen number written at the top, and we have a lot of blank space on there to write other information. The other thing that I like to do is I like to have a Sharpie or other indelible marker so that as I'm going through and looking at those sections, I can actually make a circle up there on the slide, so I will know that this is the section that I use to definitively classify this gonad. And if it's a section that I want to go back and take a picture later on, that will help me do that. And finally, if you're classifying the gonads as you're going along, you can actually write the classification on the slide itself. So this would be a slide that indicates it's immature female and it has stage 2 oocytes. Anyways, we're going to talk about the process for classifying these gonads today. Okay, so the work plan in examining these gonadal sections starts out with examining all sections at around 4x or 10x lenses, so 40x to 100x total magnification. We then are going to determine whether the tissue we're looking at is gonad or not gonad, because as it turns out, sometimes you will end up over here. That is, you've removed a tissue from the fish that is something other than gonad. In that case, we're just going to stop and we're going to remove that specimen from our histology database. On the other hand, if that specimen is gonad, we then have one or two different ways we want to go. If the gonad appears to be ovarian, we can continue to examine it at 100x total magnification. However, if it appears to be testis, we need to use 400x or greater total magnification in order to be able to distinguish whether that gonad is mature or immature. Okay, so there are basically 3 parts we're going to go through today. In part 1, we're going to talk about the classification of ovaries. In part 2, we're going to talk about the classification of testes. And in part 3, we will talk about the classification and identification of other non-gonadal tissues. All right, let's start out with the classification of ovaries. Ovaries are the female gonads, and oocytes, or eggs, are their gametes. Now, ovaries are classified as mature or immature based on the stages of oocytes, or eggs, that are present. And in general, we classify these ovaries by the most advanced or most developed oocyte class that is present. And this is important because many coral reef fishes have what we say are group synchronous oocyte development, meaning we might see several different stages of oocytes within the ovary, but we're only classifying that ovary based on the most advanced stage. All right, let's take a look at the anatomy of an ovary. The outside of the ovary is surrounded by an ovarian wall, which is made up of fibrous connective tissue as well as smooth muscle tissue. Some authors will actually be able to decipher whether this gonad is mature or immature based on the thickness of this gonadal wall, but we do not do that. We choose just to look at the oocytes. Okay, within the ovary itself, the ovarian cortex is divided into lamellae. Lamellae are just layer-like structures, and the lamellae themselves contain the oocytes. Remember, the oocytes, which are all throughout here, may be at different stages of development, particularly if we're looking at a mature female. The oocytes are the gametes we are classifying in order to assign maturity to these specimens. And finally, on the inside of the gonad, we have something called the ovarian lumen. A lumen is just a hollow area in any organ, but this lumen is where the oocytes will be ovulated to once we go through the process of ovulation. And those ovulated oocytes will then pass out of the urogenital duct when that fish spawns. Okay, now that we've looked at some pictorial representations of ovaries, this is what a real ovary might look like in one of your histological sections. Okay, so at first glance, you can see that it's divided into different lobes, and each of these lobes is a lamella or layer in there, and that in between those lobes, we might have a hollow space, and that is going to be called our ovarian lumen. In this case, you can notice that the ovary itself is made up of oocytes that all look the same stage of development. This would be an immature female ovary that only has what we call stage 1 oocytes. So we're going to go through that process now of assigning maturity. Okay, so there are 4 different oocyte classification schemes, and there's actually many more than that. You'll find some authors that have 6 or 8 different stages. We tend to go with 4 stages, and these stages were first described by Wallace and Selman in 1981 in their paper on oocyte development. So the important part here for assigning maturity is just being able to decide the difference between these 2 stages here and these 2 stages there. Because if we have an ovary with just stage 1 or 2, it's going to be an immature ovary. On the other hand, if we have an ovary that has stage 3 and 4 as well, that would be classified as mature. I just want to emphasize again that most coral reef fishes have several different sizes and developmental stages of oocytes within their ovaries if they are mature. So we could expect a mature fish to have both stage 1, stage 2, stage 3, and possibly even stage 4 oocytes. And this is what we expect for group synchronous oocyte development. Okay, our first oocytes here are our stage 1 or primary growth oocytes. These are the smallest and darkest oocytes in a fish ovary, and they tend to be both present in immature females as well as mature females. So if we see them, we see that they have a very angular shape and appearance. They have a very dark cytoplasm, and they have a very conspicuous nucleus that is pale. It's relatively large compared to the size of the oocyte, and it may contain perinucleoli in there. So again, very dark, irregular shape, and very small, usually around 30 to 50 microns. If we take a look at these in histological sections, this is what stage 1 oocytes look like. Okay, so very dark there. Again, we see a white pale nucleus. The other thing we do not see is we do not see a shell or zona radiata surrounding that egg. We're going to see that in stages 3 and beyond, and it's absent in stage 1. So it's important not that you just match pictures up when you are identifying ovaries, but you actually match up the written criteria for what determines what type of oocyte it is. These are stage 1 oocytes. Okay, stage 1 oocytes, if you zoom in, may also have the perinucleoli that I talked about, and then also they might have this sort of cytoplasmic inclusions around the nucleus as well. So be aware there is some variation, but the two things you want to look for is dark cytoplasm, angular appearance, and of course the lack of a zona radiata. All right, these are other examples of primary growth. You can see that the entire ovary is primary growth oocytes. We would call that an immature individual. Same here. This is another example of an immature female. It looks different now because this is from a parrotfish and they have elliptical oocytes. But once again, the oocytes are dark staining and they have a circular nucleus, which makes up a large proportion of that oocyte. Now let's move on to stage 2 oocytes. Stage 2 oocytes are a little bit larger. You'll notice that they're more circular, and they begin to accumulate some lipid vesicles or lipid droplets. So we call it the yolk vesicle or lipid vesicle stage. Now, lipid does not stain with any of the common stains used in histology. So it does not stain with eosin or with toluidine blue. So these lipid vesicles should appear clear in the histological section. So here you can see a stage 2 oocyte that's just beginning to form these lipid vesicles on the periphery of the nucleus. Any individual with a stage 2 and stage 1 is still considered immature unless it has more advanced stages. Here are other examples of stage 2 oocytes. You can see out here we have stage 1s. You can see they're small, they're angular. Stage 2s have gotten larger. They have a more circular appearance, and we can see those vesicles that are appearing around the nucleus. Again, those vesicles are filled with lipid or oil, triglyceride, but stage 2 oocytes do not allow us to classify that individual as mature unless there are other oocytes more advanced than that. Other examples of stage 2. Again, the ovary contains lots of stage 1 in here, and the stage 1s are 30 to 50 microns on average, and then we have something around 100 microns or so, maybe 150, and this is a stage 2 oocyte, and it has those lipid vesicles and the circular appearance. It's also less dark staining than the stage 1 oocytes. So just in summary, stage 1 and stage 2 oocytes may be present in either mature or immature females, but if we only see stage 1 or 2, we classify that female as immature. Okay, more examples of stage 2 in here as well. Okay, so you see stage 1 and 2 along the ovarian lamellae. All right, let's talk about stage 3. Stage 3 is vitellogenesis. Now, vitellogenesis is the first stage that we begin to classify that female as reproductively mature. Now, vitellogenesis is the accumulation of yolk protein called vitellin, and this vitellin is synthesized in the liver and then is transported to the eggs itself, and there it becomes vitellogenin. And the point at which those oocytes take up vitellogenin is the demarcation between mature and immature. So in vitellogenic oocytes, we see these yolk globules or granules, and they are made up of the vitellogenin protein. We will also still see the lipid droplets that we saw before. And the other thing we'll see is a conspicuous shell to this egg, and this shell may be called the zona pellucida or zona radiata. So here is an example of a stage 3 oocyte. So what can we see? First of all, we have the nucleus here, or germinal vesicle. Beyond that, the white staining areas are the lipid that began to appear during the lipid vesicle stage. And then out at the periphery, we see our yolk globules or yolk granules, and these are made up of vitellogenin, and they occur in the stage 3 oocyte. The next thing we see out here is our zona radiata or zona pellucida. This is the shell of the egg, And we generally do not see this on stage 1s, usually not on stage 2s either. So it's one of the criteria we can use to help identify stage 3s. Okay, here is a 400x micrograph of a stage 3 oocyte. And on the outside, we can see the follicular epithelium. The follicular cells are epithelial cells that surround and nurture the oocyte and produce hormones. Beneath that, we can see our zona radiata or zona pellucida, and that is this thick acellular protein membrane surrounding the oocyte. We generally just see this in late stage 2 and stage 3 and 4 oocytes. So it's one of the criteria we can use to help recognize these oocyte stages. Okay, more examples of stage 3 oocytes right here. You can see that there's a wide variation in size. This is early stage 3. We have accumulation of the protein granules, and this is later stage 3. So they can double or even triple in size during stage 3. But the 3 criteria we're looking for are the yolk globules, the vitellogenin. We're looking for the zona radiata, and that's probably about it, okay? Okay, realizing that oocyte stages look different in different species. This is a parrotfish once again, and it has elliptical oocytes, but we still see the conspicuous zona radiata, the shell. We see that it has lots and lots of yolk granules, that protein that stains with the hematoxylin eosin, or it will stain with our toluidine blue, and it also has some lipid vesicles in there as well. Finally, identifying early stage 3s from late stage 2s can be a little bit difficult, but here's a slide that shows the difference. Here is a stage 2 oocyte. You can see the accumulation of the pale white staining lipid vesicles, but you do not see any yolk granules. Moving up to stage 3, we see larger lipid vesicles here, the nucleus, and then at the periphery of the oocyte, you begin to see these small, darker staining vitelline granules. So this is vitellogenesis. The other thing is, notice the outside of the oocyte here has a conspicuous zona radiata in the early stage 3, and that is missing in the late stage 2. So once you've looked at a few of these, you can get pretty good at distinguishing stage 2 from 3. All right, now let's go on to our final stage, which is maturation. So maturation is a little bit rare to catch in an ovary because it can happen very quickly. But during this process, the vitelline, the yolk protein, is going to coalesce into larger globules or even form large sheets in there. The other thing that'll happen is we will have coalescence of the lipids into a larger lipid droplet. And we might even have the cytoplasm becoming more uniform in appearance as that oocyte begins to enlarge and hydrate during final oocyte maturation. So let's look at our first 4 stages we talked about, our first 3 stages. So back here we had our stage 1s, down here we had stage 2s, and these are stage 3, A and B. So how big are the stage 4s in comparison? Well, pretty big. Something around this size right here, this would be what we would call an early stage 4, and this might be a late stage 4. So in early stage 4, what's happening is the yolk globules are coalescing and getting larger, and the oil droplets are also coalescing and getting larger. By the time we reach final oocyte maturation, or stage 4B, we have a very large oocyte, and it also has an irregular border because it's taken on water during hydration. And when we dehydrate that in the process of our histological processing, the alcohol draws the water out and causes this cell to shrink or crenate, which gives us a starburst appearance. Okay, so here are some examples of early stage 4 oocytes. For comparison, up here is a stage 1. Here is a stage 3 vitellogenic oocyte. Here is an early stage 4. You can see it's much larger and that the oil droplets have gotten larger and coalesced. And also the yolk granules have gotten larger and coalesced as well. We still don't see the sheet vitelline that we see in some species as they undergo final maturation, but we would call this a mature stage 4 oocyte. Okay, these oocytes show the sort of irregular appearance that happens during hydration after we put those oocytes into alcohol. You can see that the ooplasm is uniformly staining with this sheet vitelline here, and that the membranes itself are very convoluted as a result of crenating during the dehydration process with alcohol. So very characteristic of stage 4 oocytes. Another example of stage 4, early stage 4 in there, we're getting conglomeration of the lipid droplets into large droplet, and then we're getting an amalgamation or composite of all that yolk protein. So this is stage 4. In comparison, stage 3 up there. Okay, finally, this is a very characteristic stage 4, late stage 4. Again, we can see that we have these irregularly shaped oocytes, and this is an artifact of our processing. They were very giant, large oocytes. If this was a live fish and you squeezed its abdomen, they would pop out and be nice and clear. But when these oocytes undergo histological processing, they shrink in the alcohol and they give us this weird appearance there that indicates stage 4 oocytes. Okay, last but not least, there is variation among species. This is a species of lanternfish that does not go through the same exact parameters we were discussing in the previous slide. It does have coalescence of the main lipid droplet and coalescence of the yolk granules, but it does not show the same star-shaped appearance. But at the same time, it undergoes final oocyte maturation in a similar way. Okay, last but not least, before we have our quiz or our assessment, we need to talk about postovulatory follicles. Now, you've heard of follicle before as being something like in a hair. A follicle is a sac of epithelial tissue that surrounds and secretes something. In this case, these follicular cells surround the egg and nurture it as it develops. And once that egg is ovulated, what is left behind is something called a postovulatory follicle. In humans, we would call this a corpus luteum. But in fish, we just call it a postovulatory follicle. And this is important because it indicates previous recent spawning, particularly if that follicle is still open, then it indicates very recent spawning, whereas if it's closed, it indicates spawning within the last 6 to 12 hours. So this is an example of an ovary with postovulatory follicles. You can see right here, we have stage 1 oocytes, and then we have follicles that are missing the oocyte. The oocyte has been ovulated, and these are open follicles, which indicates very recent ovulation. So indicates that we have recent ovulation, and that is definitely a mature female. Other oocytes we can see in there, that looks like a 3 in there. It might be a 2, can't tell from right here, but it's definitely a mature ovary if we see postovulatory follicles. Here's another example of a POF, or postovulatory follicle, in a species that has already shrunk up the follicle, and what we see now is a closed postovulatory follicle. Again, it still indicates recent spawning, but not as recent as looking at a follicle that's open. In any case, we would classify this female as mature because of the postovulatory follicles, but also because she has stage 3 oocytes here, or vitellogenic oocytes. Okay, we're now at the point where you've completed the first module on the anatomy and development of the ovary and oocytes. So we're at a point where Dindy is going to initiate a poll, and we're going to look at 5 different gonad sections from ovaries. And for those 5 ovaries, you're going to rank them as stage 1, stage 2, stage 3, or stage 4. So we're going to look at all 5 together, and I will move through them at probably 30-second intervals. And you want to click in the answers, and when we're done, we'll take a look and see how you scored.
Silahkan Bapak Ibu memilih jawaban yang ada di polling. Kalau Bapak Ibu mengklik ada tulisan polls di bagian bawah layar, itu ada pertanyaannya. Ini untuk pertanyaan pertama, silahkan pilih multiple choice.
Okay, so this is gonad number 1. So please rate gonad number 1 at stage 1, 2, 3, or 4.
Ross, can I jump in and point out that they can move the poll out of the way if it's blocking the picture of the slide? So you can just drag that poll, the questions to the side.
Ya, I forgot to mention, Bapak Ibu, jika gambarnya mungkin tertutup oleh kotak polling, bisa digeser.
Just mark right there. Okay, now let's go on to practice ovary number 2. Again, classify this ovary as stage 1, stage 2, stage 3, or stage 4. Remember, we're always going by the most advanced oocyte stage present. All right. In that case, let's go on to ovary number 3. So for number 3, you once again want to classify the latest stage of oocytes present as stage 1, 2, 3, or 4. Keep in mind that you can scroll down over here as you enter your answers. So you should see all 5 answer places to answer. Okay, let's now go on to ovary practice question number 4. Once again, classify this ovary as stage 1, 2, 3, or 4 based on the most advanced oocyte stage. All right, time for our last question. Question number 5. Classify this ovarian section based on the most advanced oocyte stage present. Again, the stages are 1, 2, 3, or 4. Okay, hopefully everybody at this point has had a chance to vote. So if Dindy could go ahead and publish the results, we'll go back through these 5 questions and talk about what the right answers are. Okay, I'm just going to move those out of the way. And again, you can move those out of your way. You can drag them. So let me drag those so I can see myself. Okay, so question number 1. We had a variety of answers ranging from 1 to 4. The most frequently chosen answer is the answer that's correct. So these are stage 3 oocytes. If you take a look at the oocytes themselves, they have a zona radiata there. Okay, they have a shell. They also have lipid vesicles, which are in stage 2 and beyond, but they also have yolk granules. Particularly if you look down here, you can see the yolk granules that are between the shell and the nucleus. So this would be a stage 3 oocyte, and we would classify a female with these oocytes as mature. Okay, question number 2, more people got this right. So the majority of people chose stage 1 oocytes. So stage 1 is over here, okay? And that is exactly what this ovary contains. Lots of stage 1 oocytes. It's zoomed out so you can't see the nucleus, but they are all angular, dark staining oocytes. They do not have a shell or a zona pellucida, zona radiata, and so they are stage 1 primary growth oocytes, and we would classify this female as immature or inactive. Okay, let's look at practice question number 3. Can we scroll down in the results a little bit? Or do we have to do that individually? Okay. So practice question number 3, many people got this correct. So the most of you chose stage 4, and that is exactly correct. So these are stage 4 oocytes. They have lots and lots of vitelline in there. They had this irregular star-shaped appearance because they were so large that when they hit the alcohol during infiltration, they shrank up as a result of osmosis. So we have this starburst-shaped appearance. So stage 4 is correct. All right, for question 4, the correct answer here should be stage 2. So this is a stage 2 oocyte, our cortical vesicle, or excuse me, yolk vesicle stage. It has the lipid droplets that are present. It does not have a conspicuous shell, but it is larger and more circular than our stage 1 oocytes that are here. But because this ovary just contains stage 1 and stage 2, we would classify it as immature. Okay, and finally, question number 5. The correct answer to question 5 would be stage 3. These are stage 3 oocytes. They have lots and lots of vitelline granules in there, and they have a very conspicuous shell or zona radiata. So this would be classified as a stage 3 oocyte, and it would be a mature ovary because anything beyond stage 3 is considered mature. Okay, I think that's it for the survey. Now, do we have time for any questions?
Yeah, so there are several people raising their hands, and I will choose 3 people for this question session. First, Deli Matruti. Please, Deli Matruti. Deli Matruti. Bisa tolong di... Okay, I think she or he lowered the hand. I will choose Denny Suhernawan. Denny Suhernawan, please. Yeah.
Can you hear me, please?
Yeah.
Okay, thank you for time to ask about the question. Okay, I show that in a slide that we found that some oocyte at different stages, and then how to determine the stage of the ovary. Sometimes we found stage 1 oocyte and stage 3, and then how to decide the stage of the ovary. Thank you very much.
Sure. It depends on what you're using the data for. In our cases, we're just using the data to estimate the size of maturity for the fish, or the L50. So in that case, even though it was stage 1 and 2, if it had even just a few stage 3, we would call that a mature female for the purposes of estimating L50. On the other hand, if you want to use that ovary to estimate spawning seasonality, you would need to look more at the area of the ovary that is occupied by vitellogenic oocytes or mature oocytes. But that's not what we're doing today. But you certainly could do that to get a little bit more estimate of reproductive output and seasonality.
Thank you, Ross. So the next person that I'm randomly check is Indrastiwi PR. Indrastiwi, silahkan. Ibu Indrastiwi.
Yes. Can you hear my voice?
Yes.
Yes.
Please go ahead.
Okay, thank you. I will ask about the post stage. If an oocyte is found, can the follicle or pulp regenerate into stage 1 again? Thank you.
That is a good question. I don't think the follicle will regenerate a new oocyte. Usually that follicle will degrade. And sometimes if the oocyte is not ovulated, it will degrade as well, become something we would call yellow body or melanophage, macrophage center. But I'm not aware of a follicle being able to generate new oocytes. The oocytes themselves are generated by mitosis and meiosis through our oogonia, which are part of the epithelium of the ovary.
Thank you, Ross. The next person is Reni Tumbo. Reni Tumbo, please go ahead.
Okay, thank you. Can you hear me?
Yes.
All right. My question is, what are the distinct differences between species on the base of the oocyte maturation?
What do you mean spaces? Sorry.
I mean the variation between species. Are there any distinct differences on the oocyte stages of maturation? Are there?
Species, Ross.
Species. Oh, okay, sure. Yes, species.
Sorry.
Absolutely. Absolutely, there's variation. And I'll talk about in our next meeting that when you use the SOP, realize that your oocytes, if it's a new species, might look very different. And so it's important not just to match pictures, but use the written criteria. Are there yolk granules? Then maybe that's stage 3. Yeah. Lots of species vary.
All right. Thank you.
Yeah. Sure. Next person is Sampari Suruan. Sampari Suruan, please. Sampari Suruan. Sampari. Oke, saya lengkap ya. Suryadi Saputra, silahkan Pak Suryadi Saputra.
Okay, thank you. Do you hear my voice?
Yes.
Okay. Thank you. We have two experience with cobia fish, about 12 kilos each fish. One experience after we injection HCG, human choriot hormone, until today, the female we injection is there. Another experiment, we have a fish female is there too. After we give another supplement about 2 and 3 days is there too. My question is, supplement or hormone that will be that cobia fish. Thank you.
Okay, Ken, you might have to help with this one. So you injected the fish with either chorionic gonadotropin or something like LH, luteinizing hormone, to induce ovulation. Is that correct?
After we selected a female, we injection HCG, human choriot, not lutein choriot, human choriot hormone.
Yeah, unfortunately, we are not aquaculture or endocrine specialists either. So really wouldn't know what led to the mortality of the fish. I do know that, you know, usually LH or something like that will cause ovulation, but yeah, I wouldn't know the cause of the mortality. Ken, can you chime in?
I'm sorry, I don't have anything to add.
Yeah. This is connecting about oocytes, TKG4. After we selected this female, so for this I injected, but it's okay. Thank you for your answer.
Terima kasih Pak Suryadi Saputra. So I think now we have reached the next presentation of Ross on identifying male gonads. Ross, time yours.
Okay, give me just a second to load that presentation up. Okay, can you all still hear me?
Yep.
All right, so now we are moving on to the second part of the presentation, which is classification of testes. Now, testes, of course, are the male gonads. They are bilobed, just like the female ovaries, but the gametes here are spermatozoa, not oocytes. And so unlike the ovaries where we had to classify the gametes in 4 or 5 stages, we really only have to ask one question with ovaries. Do we see spermatozoa that have tails, or do we not see spermatozoa? So in this case, for the estimation of L50, we just look at the testes to determine whether they have spermatozoa. If they have spermatozoa, we classify that testis as mature. If they do not have spermatozoa, we classify it as immature. Now, it's important to do this is to have very thin sections in order to see the spermatozoa, but also have a high power 40x lens, or ideally a 100x oil immersion lens. And so we will be taking a look at male gonads at both of these magnifications in order to assign maturity. Before we do that, we need to talk about the organization and anatomy of the teleos testis. So the testis of most marine fishes is divided into lobules, which we'll talk about. So these lobules are sort of blind-ending tubes that may connect with other tubes. And the lobules have a central lumen that we'll talk about as well. And just like the ovaries, we have a gonad wall comprised of fibrous connective tissue and also some muscle tissue. And we may have a central sperm duct located usually on the medial side of either testis. So this shows a greater sort of 400x view of the different structures within the testis. The functional unit of a fish testis, in this case, is called a tubule, or more often a lobule. So the lobule here, again, is a hollow tube consisting of an outer cortex of testicular tissue and an inner lumen. The lumen, again, is a cavity where the sperm will leave when they go through the process of spermiation. Okay, the next thing I want to point out is in the lobule on the wall is our spermatogenic tissue. And spermatogenic tissue occurs at a few different stages. We have something called spermatogonia, which basically give rise to all the other cells. And these cells become encapsulated by a Sertoli cell, and we call them a spermatocyst. It's not really a real developmental word, but we use it to denote any type of spermatogenic cells that are encapsulated. Now, early spermatocysts will have larger nuclei of usually primary spermatocytes, and later spermatocysts will have smaller nuclei as we've had more cellular division in here. It's not essential to memorize the different names for what's going on, but realize as the nuclei get smaller, we're getting more advanced in the development of the spermatocysts. Finally, remember the lobule lumen is the place where the sperm are going to enter once they go through the process of spermiation. At this point, they will have tails and they will be let go from the spermatocyst and enter that lumen and eventually be transmitted to the sperm duct. Okay, let's take a look at the anatomy of a testis, and this is of a surgeonfish, Acanthurus lineatus. And what we can see here are lots and lots and lots of lobules. There's a lobule, and there's a lobule, and the lobule has a central lumen. And the big hint I'm going to give you for looking at testes is look for open space. Open space is where you're more likely to see spermatozoa and be able to identify it. So a testis with lots of open space is one I would suspect of being reproductively mature. So we put some labels on the different structures in here. Again, we've got these lobules, and the lobules are made up on the sides of spermatocysts, and the spermatocysts contain our dividing cells, our spermatogenic tissue. And then on the internal portion of these lumen, we can actually start to see some of the tailed spermatozoa. We're not at a great magnification to see that now, but hopefully I will show you in a minute at higher magnification. Now, testis anatomy can look different depending on the orientation of the knife or cutting surface to the gonad as we cut it. So here you can see those lobules more in an oblique or even longitudinal section. And you can see the lumen of those lobules is quite long, and it contains this fuzzy material in here, which is actually spermatozoa. Again, if I look at a gonad like this at 100x and I see lots of open spaces with some fuzzy stuff in the middle, I want to look harder because chances are that is a mature male with spermatozoa. Okay, here is a 400x magnification of a longitudinal view of some of those lobules. Now, what you'll notice now is there's different colors going on, and that is a phenomenon that happens with the stain that we use, is that sometimes, particularly when that stain is aqueous, it will stain the spermatozoa different color than the surrounding spermatogenic tissue. So in this case, it's very easy to identify the spermatozoa because they appear with different colors there than the surrounding spermatogenic tissue, which is a lot darker and a lot bluer. So this would be a testis of a mature male, but again, we need to view it at 400x in order to be able to assign maturity. Okay, now we're going to go through some slides and classify them as mature and immature. The first ones we're looking at both 100x and 400x. And for a lot of small species or small individuals, we actually collect more than just the gonads. We collect all the tissue that's in the dorsal body cavity. So here we can see at 100x, we have a testis, we have a swim bladder, and on the left side, we actually have the muscular body wall. If we look at that initially, that testis, we do not see any open space. It's very compact, a very tightly packed spermatogenic material. When we zoom in at 400x, that's still the case. Lots of tightly packed spermatogenic tissue, no visible spermatozoa, no open spaces. We would classify this as an immature male. Okay, here's another example, and this is from a large individual. We removed the testis. We expected it to be mature based on the testis size, but that is not what the histology showed. Once again, the 100x view shows lots and lots of tightly packed lobules, and this tissue in between here is going to be some of that fibrous connective tissue. Out in these spaces is where the Leydig cells are that secrete the hormone testosterone. And if we look at the 400x view, we still see lots of lobules with tightly packed spermatogenic tissue, but it all seems to be the same stage. We see no open spaces, no conspicuous lumen, and so this would be called an immature male. Now let's look at another species. This is actually another one of those, I think it was a lanternfish. And again, the testis at 100x is actually quite small. We can identify the testicular tissue by the presence of spermatocysts or encapsulated spermatocytes. And if we look at that at a higher magnification, we continue to see the spermatogenic tissue here, but we do not see any spermatozoa. We do see that the nuclei are of various sizes, indicating some development, but because we see no spermatozoa, we classify this testis as immature. Okay, last immature male. Once again, we look at the 100x view, tightly packed tubules or lobules, no open spaces, and at 400x, we again see tightly packed spermatogenic tissue, no open spaces, do not see a lumen, and we do not see any tailed spermatozoa. So once again, we would classify this as an immature or inactive male. Now let's take a look at some mature males. So this is the twin testes of a mature male at 100x. And what you can see first is lots and lots of open space with fuzzy material in there. That fuzzy material makes us think this must be spermatozoa, but to confirm that, we have to be at 400x magnification. Here's what it might look like at 400x magnification. So what we see here is a lumen to a lobule, and we see lots of heads here of spermatozoa, and this is what we're looking for right here. This fuzzy material, these are the tails. And usually all the tails will point in one direction, and that indicates for sure that we have a mature male because we have these cysts of spermatozoa that are present within the testes. So definitely mature male. Okay. Here's another example of a mature male, again at 400x. You can see up here is the lumen to a lobule, and this would be the cortex to the lobule. But what you can see right here within even the cortex of that lobule is we have this spermatocyst that has these tails in here, tailed spermatozoa. They're still retained in the wall of the lobule, and they have not yet gone through the process of spermiation or being released. But once again, we would classify this as a stage, as a mature male. Okay, another thing we can do with our stains, since we only use one stain, is if we use toluidine blue and then we place some water on those stain sections as we observe them and cover slip them, we can get an added contrast agent that's not normally present. So normally with toluidine blue, you just see lots of blue, but if you add some water in a cover slip, you begin to see blues and purples. And so this is a testis at 100x. You can see different colors in there. And then if we add 400x, you can see that the lobules themselves have these spermatocysts in there, and that pink material in there are the tails of our spermatozoa. So this would be a mature male because it has spermatozoa in there. And again, we classify it as mature. But once again, you have to use your 40x objective in order to identify that. Okay, last but not least, we have a testis of another lanternfish, a myctophid. And if you look at this page here on the right, you can see that there's some open spaces, there's some visible lobules. And if you look at 400x, it looks different. We don't see the tails in there. As it turns out, these comma-shaped structures that you can see are actually the spermatozoa of these lanternfish. They do have tails, but they're only visible under electron microscopy. So all we can see here is the comma-shaped heads to the spermatozoa. But just keep in mind that, like with ovaries, there can be some variation in the anatomy of the testes and the anatomy of the gametes themselves. Okay, last but not least, in section 2B, I want to talk about something called ovotestes. Now, ovo means ovary, and testis, of course, is a testis. So ovotestes are gonads which contain both ovarian and spermatogenic tissue. Now, ovotestes can indicate lots of different things. We can see ovotestes in individuals that have undergone environmental stress. We can see ovotestes in individuals that differentiate from a bisexual gonad, but themselves are not sex changers. And of course, we can see ovotestes in individuals and species that change sex. And so it's very important that you don't just assume that your species is a sex changer the first time you see an ovotestis. Okay, so this is kind of what an ovotestis might look like. If you remember back to our first talk, we talked about ovary anatomy. And remember, the ovary was divided into lamellae, which are these finger-like structures. And that is very lamellae looking. The other thing you can see are these small, dark staining primary growth stage 1 oocytes. So that is indication of female anatomy, okay, as is the lumen right here, okay? The lumen is very indicative of a female ovary, and we do not usually have a lumen within a testis, or at least not this type of lumen. If we look closer at these lamellae, however, we actually do see spermatogenic tissue on there. So that leads us to classify it as an ovotestis. So this is what that ovotestis might look like at 400x magnification. Again, we have our primary growth stage 1 oocytes over here, okay, for sure. And then we have a spermatocysts of different stages of sperm development. We probably have primary and secondary spermatocytes in there. We do not see any spermatozoa in there, and we don't see any advanced stage oocytes. So this ovotestis would be non-functional as male or female, but again, may indicate a transition between sex or may indicate another developmental process. Okay, finally, in some testes of sex-changing species, we can see a vestige of their former ovarian architecture. This is a testis of a fish that contained copious amounts of sperm, so it was definitely functional as male, but it retained this, what we call peripheral lumen, and that is an anatomy that's very similar to what we see in functional females. And so a lot of times in secondarily derived males, males that were derived from initial females, they will retain this lumen for a small amount of time. Okay, we have completed the testis and ovotestis section of this module, and now it's time for 5 more practice questions. Now for these questions, you're going to be given 5 gonad sections of testes, and you basically have to classify them as mature male, immature male, or ovotestis. And once again, you can move that box to the side so you have a better view of the gonad.
Silahkan polling dimulai.
Okay, this is our first practice question. And again, classify this as a mature testis, immature testis, or ovotestis. Okay, let's go on to question number 2. Question 2, the same thing. Classify it as a mature testis, immature testis, or ovotestis. Okay, let's move on to practice question number 3. Once again, classify this as immature testis, mature testis, or ovotestis. Okay, question number 4. Let me move out of the way. You're at 400x. Okay, classify this as immature testis, mature testis, or ovotestis. All right, last but not least, let's move on to practice question number 5. So classify this as testis, ovotestis, immature testis, or mature testis. This is at 100x magnification. Okay, hopefully everybody has had a chance to answer the questions. Dendi, can you please close the poll and show us the results? All right, I'm going to move this out of the way so I can see myself. And once again, you can scroll down the poll as we go through it. So we're going to go back to the first section and see how everybody did. Okay, it looks like for the first question, the majority of participants chose inactive or immature male. Is that correct? I can't quite read it from here.
Yeah.
Yes, that's correct.
Okay, so that is the correct answer. And the reason we can say it's correct is if we look at the gonad, we can see these tightly packed lobules. Remember, lobules are the functional unit of the male gonad in fishes. And the lobules are tightly packed with spermatogenic tissue, but we do not see any open spaces. And we also do not see any spermatozoa. In all truth, we would actually need to examine the gonad at 400x magnification to verify this. But given only the 100x slide, I think that is a good classification. Okay, for our next slide, Ken, can you tell us the most frequently chosen answer?
For number 2, the most frequently chosen answer was mature male.
Okay, so mature male is absolutely correct. So we can see these lobules here, and they're mainly just lumen of the lobules. We see very little tissue on the outside there, and this is often what we see in a testis at the end of a spawning season. The majority of tissue there has been converted into spermatozoa, and we see copious amounts of spermatozoa within the lumen and also within the sperm ducts. So this is a very easy to classify mature male. Okay, Ken, what was the most frequent choice for question 3?
This one was a lot closer, but the most frequent choice was mature male.
Right, and this is a hard one because the sperm in there are so packed. Honestly, this is probably at 1000x magnification, so it might have thrown you off. The heads in here are the head pieces of the spermatozoa, and all the purple stuff in here are just very tightly packed tails. The tails are what differentiate spermatozoa from all the earlier stages there. So seeing lots and lots of these tightly packed tails is indicative of a mature male. Okay, question 4, what was the most common choice?
Mature male was by far the most common choice.
Absolutely, and this is an easy one. Initially, if you look at this material in here by itself, it's very tightly packed. We do not see any spermatozoa. We do not see open spaces. So we would think maybe immature, but here we look at a nice open space. We can see those heads of our spermatozoa, and then we can see the telltale tails of the spermatozoa as well. So very definitively a mature male. All right, last but not least, question number 5. What was the most common choice for this?
78% of the participants chose ovotestis.
That is an excellent result. Yes, this is an ovotestis. We see that we have oocytes, our primary growth oocytes. We have an ovarian lumen, similar to what we would in an ovary. But if you look closer in there, you can see some spermatogenic tissue as well. So this is very definitely an ovotestis. Excellent job. I think now we have some time for some questions.
Yes, great. Thank you very much, Paros. So everyone, so now we have a question session. So I will randomly choose several people. I will now choose Pak Pandey. Please, Pak Pandey.
Yes, thank you, Ibu Eni.
Sama-sama.
And thank you also for the nice and clear presentation. I would like to that we can clearly see which is immature or mature male for the fish. But can we see from the histology that this is mature but not fertile, or infertile males from the histology? Thank you.
I'm sorry, I didn't hear that last part. Can you repeat again?
Yeah. Can we see from the histology that the mature males, but not fertile or infertile male? Thank you.
Oh, that is a great question. And that is probably more of an aquaculture question. To be fertile, I would assume that you need sperm within the sperm duct and within the lumen there too. That would be a question for an aquaculture specialist rather than ourselves just saying, okay, we see spermatozoa for a fisheries standpoint, we're going to classify that as mature male. I can tell you that when we look at the sperm ducts and when we look at the sperm within the spermatocysts, they tend to correlate pretty well. That, I don't think we see a smaller size of maturity just by using the spermatocysts versus the sperm in the sperm duct. But yeah, I would assume you would have to look in the sperm duct itself to corroborate that that was a fertile fish.
Terima kasih Pak Pande. Berikutnya Ibu Shelly Patipeluhu. Ibu Shelly, silahkan.
Yeah, well, it was my question for the oocyte ovary actually, but well, I can ask you if this ovotestis, which is have ovary and testis, is that always the process of it happened because of stress, or are there any natural process to have this possibility, whether it's become male or female? Thank you.
Sure. Firstly, it is natural in sex-changing species to have an intermediate ovotestis. If we're going from ovary to testis, as in a protogynous species, in a protandrous species, we go from testis to ovary. So that would be natural. There are some species where the juveniles start out with a bisexual gonad, but only one sex of that gonad matures. So I think in some species like Dacilus, we see that where initially it's bisexual, but before the gonad matures, one part of that gonad, the ovarian part, might mature and the testicular part would atrophy. So that would be natural as well. And occasionally you'll just see an oocyte in a testis, but if you see a lot of them in there, it could indicate environmental stress, or it could indicate sex change.
Thank you, Ross. Thank you, Ibu Shelly. And sekarang Pak Fabian, silahkan Pak Fabian.
Okay. I want to ask about female section before. Now I'm still working on my thesis research about lunar cycle and reproductive activity yellowtail pussyler fish in Karimun Jawa. So in my result is GSE or gonadic somatic index peak happen in full moon phase. And I found oocyte ovulation is present, but I use a common microscope, not use histological way. But I still can look hydrated oocyte. Can that prove this fish spawning at that time when this fish hit the peak of GSI? Thank you.
Absolutely. GSI is great because it's an objective measurement. We've all done estimation of spawning seasonality using both histology and GSI. The histology supports the GSI. I find the GSI much easier to use. What do you think, Ken?
I think, well, GSI is easier to do when you go through the whole process of weighing the gonads and the fish. So it's a great place to start. I know that we're sharing something here. Ross sometimes has difficulty looking at plots of the different egg stages versus seasonality. But if you look at also the proportion of different egg stages versus season, it's again, oftentimes corroborates quite well with the GSI.
Thank you for all the answers and for the question. Terima kasih, Pak Fabian. And now we have 5 minutes bio break, and we return in 5 minutes, yeah, for the next session on identifying other structure by Ross, Pak Ross. Terima kasih, bio break kita.
Thank you.
Oke, selamat siang. Kita kembali lagi sekarang. So we are returning back to the training session, and Pak Ross will explain about identifying other structure. Pak Ross, time is yours.
Thank you, Annie. Can you hear me?
Yes.
Okay, so at the very beginning of this presentation, we talked about the order of events needed to go through to identify gonadal structures and then classify those gonads as mature or immature. And we said that if we have an ovary, we're going to look at it at 100x magnification, identify the oocytes. If we have a testis, we're going to use 400x magnification. But the question is, what happens if what we look at, we find out is not gonad? Well, we're obviously going to stop, but we're going to answer the question, why would you look at something that's not gonad? After all, you collected the gonads. Just a second. So this module is going to talk about the identification of non-gonadal tissues, because occasionally the person that's doing the dissection will inadvertently remove the wrong tissues from that fish. And this is particularly common in very small fish that are immature. It's very common to collect the wrong tissues, and in that case, somebody might macroscopically classify that as one thing, but histologically, we can definitively rule out that that tissue is not ovary or testis. So it's not really necessary for you to memorize all the non-gonadal tissues, but rather be able to understand and identify the fact that the tissue that you're looking at is not gonad, so you can throw that tissue out and then go on to your next gonadal section. Okay, so here we are, Fish Anatomy 101. And as everybody knows, fish anatomy is very diverse. This is an idealized fish model. If only all fish looked like this on the inside, but some internal anatomy. Many fish, of course, have a swim bladder for maintaining buoyancy. So that is something we can sometimes see in histology. We also have a kidney in there. I think it's a mesonephric kidney, if I remember. And this is very commonly collected instead of gonad. And when we look at that kidney, it's very easy to identify it as kidney and not something else. When you're picking out that gonad with a forceps, chances are if it's black in there, it's part of the peritoneum and probably has some kidney in there as well. So kidney is very frequently collected instead of ovary and testis. Of course, we have liver, which is up in the cranial part of the animal. And liver is quite a large organ, but we find that it rarely ends up in the histological sections because it's all the way up here, and most of the people are grabbing way back here to get those gonads. And of course, we have the gonad itself. You know, if all the gonads looked like this, there would be no trouble in identifying the gonads and collecting the right tissue. But again, it's very difficult, especially for the small immature individuals, to collect the right tissue. And you want to be sure you collect the right tissue. Otherwise, going through this process is going to be sort of a dead end. Okay, and of course we have the gut. I want to point out in here, the digestive tract, which extends from the mouth, the pharynx, the stomach, and then eventually out here to the anus. So we frequently get tissue cassettes that have gut in there as well. Okay, ideally, a dissection would look something like this. This is a nice big cod or something of that sort, where we can see the stomach in here. We can see the liver is very, very large. Up here in the dorsal body cavity, we have some dark pigment, probably going to be the swim bladder in there. And then back here, we see the gonad or the ovary. Again, if all of our fish look like this, it would be very easy to collect these tissues and there would be very few errors. But we found in our own collections and the collections of others that sometimes more than 30% of the tissues that are collected are in fact not gonadal tissues. And so if you're looking at a dissection like this is us working in Fiji, you're looking inside that fish going, gosh, what is that structure? In this case, you can see pretty clearly it's a nice big ovary, easy to collect. But sometimes we end up with something that's not as easy to collect. For surgeonfish in particular, it's very difficult with all the green guts that are in here to collect the right stuff. Now, keep in mind that gonads are one of the few structures in the body that are bilaterally symmetrical. That is, we're going to have two gonads. And so if you find two of something, chances are it can be a gonad. Other times, you'll be left pulling out the guts of some fish and trying to decipher what indeed is a gonad. Now, if that tissue is small enough, you may be able to embed and section all of it, but that is going to be very time consuming. So just pointing out that having a person that's very skilled and experienced on the dissection end can make this a much more fruitful process. So use your best person to do the dissections and collect those gonads. Okay, I want to show you one of the diagrams that's in the SOP, and it shows the approximate location of gonads in different fish families. And here we have the family Acanthuridae, our surgeon fishes, and Ken put together this nice graphic showing where the swim bladder might be, where the guts are, there's a lot of them, and then where the gonads are. Because as we found out through experience, the gonads in these fishes aren't where you would expect them to be. And if you look back here, there's something that looks very, very gonad-like, and Dr. Franklin knows all about this. And this is a fat body that is basically occurs in many species of surgeon fishes. It looks identical to a testis, but it is not a testis. In fact, after embedding some 50 or 60 of these and sectioning them, we later came across this paper by Fishelson et al. That describes the anatomy of this fat body. So up here you can see the testis, and back is the fat body. And the fat body is bilobed, just like a gonad would be. And regardless of whether it's male or female, we still see this bilobed structure. This is not a gonad. This is a fat body. The gonad is right here. So it's very easy to collect the wrong tissue. Well, what would that look like if we collected it? First of all, the first key that you might have collected the wrong tissue is when you go to preserve this in a fixative, such as formaldehyde, it will probably float. That's because it's fat. But then if we look at it under histology, we can see something like this. So fat tissue is made up of adipocytes. And if we zoom in to 400x, that's what we see. These large cells that are just filled with lipid or triglyceride, and the nuclei are actually on the periphery of the cells. So this is fat tissue. And because fat tissue does not mix well with water, and our plastic medium, embedding medium, is water soluble, oftentimes we get poor infiltration. And in this cases, we get a ripping and tearing of the gonad sections. So the second indicator that you might have collected the wrong specimen or wrong tissue might be when you're on the microtome and you're cutting and it's just falling apart. Oftentimes that indicates that we're cutting adipose tissue, which is not gonad. So it's a very common mistake. Adipose looks macroscopically like a testis. And so I've made this mistake and others have made it as well. Okay. So let's take a histological look on the inside of a fish. If you have the opportunity to do histology, I would urge you to use your smallest individuals and try to embed at least the large part of the viscera of the whole fish. That can give you an orientation of the anatomy of your fish histologically and help guide your future collection of gonads. So what you can see here is we have the inside of the fish, and then up above, we have our kidney, our swim bladder, our body wall, and our gonads are suspended from the swim bladder, and these are ovaries right here. They have a conspicuous internal lumen, so that is ovary. And then below that, we have the gut or the digestive tract. So if we look over here, this is a little bit further, I think, forward, and that same fish, gonads here, part of the swim bladder up there, and then the gut down here. So doing this type of embedding can help you to identify the gonads when they're present, and also help to guide your collection of gonadal tissue when you do future dissections. Okay, so what kinds of things do we find inside the fish or inside those gonads that aren't really gonads at all? One of the most frequently types of tissue that we get inside of our cassettes, if it's not gonad, will be digestive tract. This is part of the stomach, sometimes the intestines, but it's all GI tract. It has a conspicuous lumen, just like an ovary might, but it also has different tissue layers. It has an intermucosal layer, a submucosal layer, and then we have a muscularis and serosa layer. You don't need to memorize the layers. You just need to know that this is digestive tract, it's gut, it's not what we're looking for. Okay, this is also digestive tract, probably intestine, and we're looking at it in a longitudinal section. Here you can see we have a central lumen. At first it might look like an ovary. These might look like lamellae, but they're not. These are finger-like appendages coming off the GI tract there. If we look under high magnification, you can see these little finger-like villi projections, and you can see the mucosal layer right here of columnar epithelium. So it looks like an ovary, but there's no oocytes, it's digestive tract. And then we have the stomach. It's uncommon, but sometimes somebody will grab the stomach because it's large, it's turgid, it looks like an ovary, but there's only one stomach. So this is the time where a student embedded the stomach and we sectioned through it, and you can see that this is a fish that just ate all kinds of invertebrates and things like that. You can see the different color in here that indicates different types of tissues. And then something over here, if anybody can tell me what that is, I would be grateful. Just by the shape, I don't know, coccolithophorid, foraminifera, whatever. The point is, it's not gonad, so we don't classify it. We just move on. Okay, other structures we might see, we already pointed out those adipose tissues before. So there's adipose tissue near the gonads. There's also adipose tissue around the liver, adipose tissue within the omenta as well. And then there's muscle tissue. A lot of times when you grab in that body cavity with a forceps, you will pull off some membrane, which may have some muscle tissue in there, okay? If we zoom in on that, we might be able to see striations. Striations in muscle tissue indicate that it's usually going to be skeletal muscle. So skeletal muscle is striated, so we know that skeletal muscle fibers there. And then if we're looking at high magnification, it's possible you might find some smooth muscle, as in the walls of the ovary or walls of the testis itself. We might have smooth muscle, also have smooth muscle within the GI tract. And then blood. Keep in mind that blood of fish have nucleated red blood cells. And initially, when you're looking at them, they might look like spermatogenic tissue, but it's not. If you can see a surrounding to that nucleus in there and identify it as a nucleated red blood cell rather than being some kind of spermatogenic tissue. So that's a common misclassification that happens as well. And finally, kidney is also commonly collected as something that would be gonad. Again, the kidney is located in the dorsal body cavity, but it's made of a different type of epithelium. Here we can see all that sort of cuboidal epithelium makes up the tubules there. Okay. More cuboidal epithelium in there. And if we zoom in there, we can actually see the functional units there, a glomerulus within one of those kidneys. And this is all part of the tubules of the nephron. This would be the Bowman's capsule, etc. This is not gonad. So just identify it as something else and pass along. Okay, the next thing I want to say that even though when we collect the correct organs or correct tissues, keep in mind that organs don't occur by themselves. This is a beautiful testis right here, but it also is attached to adipose tissue, and it also has blood vessels above it. So initially, if you were to look at this section at 400x, you might be looking right here at the fat tissue, and you might decide, oh, that's not a gonad. That's why it's important to examine all the sections at first at a lower power, such as with the 10x or 4x objective, to identify the area where the gonad might be located. Okay, last but not least, I just want to point out a few other structures you might see within your tissue sections. For example, one of the things we do with our smallest fish is we actually cut off the top part of the fish specimen, the bottom part, and we actually take the whole visceral cavity and put it into the resin and embed it, if it's small enough to allow that. The good thing about resin versus wax is that you can actually section undecalcified bone in there as long as it's not too big. So don't be surprised if you see some bone tissue in there, okay, which is a type of connective tissue, or nervous tissue in there as well. Okay, some of the nerves in some of these fish are quite big. This is the spinal cord here. We even can see cartilage and section cartilage down there. Cartilage turns out kind of purplish with a toluidine blue in there, but keep in mind that none of these tissues are gonadal tissue. So if we see them in the section and there's no other gonadal tissue there, we would discard that specimen and remove it from our database. Okay, so we've reached the end of the sort of other category for today's modules, and now we're going to go through 10 questions, that is 2 groups of 5, and for each one of these questions, you're going to classify it as a mature ovary, immature ovary, mature testis, immature testis, ovotestis, or you're going to say it's not gonad. So Dendi, can we start the first question, please? Okay, once again, I'm just going to move this out of the way.
Pak Ibu, polling dimulai Bapak Ibu.
Okay, for question 1, identify this gonad as one of those choices visible at right. Okay, moving on to question 2. Same question. What type of tissue is visible? Is this mature male, immature male, mature female, immature female, ovotestis, or not gonad? Okay, next question. Question number 3. Classify it again as one of the choices that are visible on your screen there. Okay, moving along to question number 4. Same question. Identify this tissue as one of the 5 choices visible on your screen. Okay, moving along to question 5. And if we can move that question box to the other side of the screen, that would be helpful. So here we have a gonad at both 100x magnification on your left and 400x magnification on your right. So identify this gonad as one of the choices visible in the menu. Okay, now we'll move along to question number 6. So if we can move those menus once again. All right, so this is a 100x magnification view of something we collected from the fish. So once again, identify it as mature ovary, immature ovary, mature testis, immature testis, etc. Okay, let's move along to question number 7. Question 7, this is a 400x view of some tissue removed from a fish. So identify what that tissue is. Okay, question number 8. Same question. We've got something we've removed from a fish. The question is it gonad? Is it ovary? Is it testis? What is it? So use the menu at your right and identify that structure. Okay, question 9. Same question. Classify this structure as ovary, testis, ovotestis, or not gonad. Okay, and the last question here is question number 10. Question 10, identify this structure. We're looking at 400x magnification. Classify it using the menu at your right. Okay, Dindy, can you go ahead and close the poll for us, please? Right, so I'm going to back up. If we just move that to one side, and if Ken, if you can read the results off as I get to that question.
Sure.
Okay.
So most people chose this as a mature female.
Okay, so that is the correct. This is a mature female. I can see that, yeah, the majority of people chose that. And the oocytes that we see here are going to be, whoops, pointing the wrong way. The oocytes we see here are stage 3 oocytes. They have a very conspicuous follicle around there, and then they have lots of vitelline granules in there. So definitely going to be a stage 3 oocyte. So in that case, we say it's a mature female. Okay, question 2.
Oh, sorry. 71% of people chose this as not gonad.
Okay, so 71% of you were correct. This is indeed not gonad. This is adipose tissue, right? This is from one of those fat bodies we had inadvertently grabbed thinking it was testis. So fat under the microscope looks a lot different than ovaries. These are all just big fat adipocytes. They don't stain with hematoxylin. They don't stain with toluidine blue because fat doesn't stain with those dyes. So this is a good answer, not gonad. Question 3.
This was a close one, Ross. 65% of people said it was testis. 33% said it was an immature male, whereas 32% said it was a mature male.
Okay. And again, I see the reason for the different choices. In classifying a male, you look at it at both a low power, say 100x magnification, and a high power 400x. This is just the 400x. So maybe it's hard to understand that this was a lobule at one time. And again, this is all tightly packed spermatozoa. It might be hard to see without the 1000x, but just based on our experience and our classification of this gonad, when you see a lot of white open space and you see a lot of fuzzy things in there, you want to zoom in because chances are that is sperm. In this case, will be a mature male. Okay, question 4.
56% of people said this was not gonad.
And that's the best choice. This here is, again, is adipose tissue. It's zoomed in at 400x. We can see the large adipocytes. Up top, we see the tearing, and we have tearing because adipose tissue does not embed well with our water-soluble plastic embedding media. So this is not gonad. Number 5.
48% thought this was immature male.
Immature male is correct. Can we move that box over to the left? Whoops. So what you can see here is, again, at 100x view, tightly packed tubules or lobules, no internal space there. And that view continues at the 100 or 400x. We don't see any internal luminal space. We don't see any spermatozoa. So this is an immature male. Okay, what did they choose for question 6?
Question 6, most people, 48%, said this was an immature female.
Yeah, immature female is correct. This is definitely an ovary. And here we have the ovigerous lamellae where the eggs are made. And you can see that all the eggs, the oocytes, are dark staining. They're irregular shaped. They're small. So they're all stage 1. There might be a stage 2 in there, but they look to be stage 1. So we say that it's an immature female. Number 7.
7, most people, 48%, chose mature male.
Right, and that's correct. So we're zoomed in at 400x. And again, look for the little tiny heads here, the heads of our spermatozoa, and the tails sometimes are so tightly packed, you can't make out the difference between those tails. So these are spermatozoa tails here, and there's some tails down there. I like to think of it as like a bouquet of flowers, except instead of flowers, it's spermatozoa, and the tails are just like the stems of those flowers. So this is a mature male. Okay, question number 8.
72% chose not gonad.
That is correct. So this is definitely not gonad. Whoops. This is definitely digestive tract or GI tract. It's probably intestine in there. We can see the finger-like projections coming off the outside of the GI tract. So we know that this is not gonad. Question 9.
Most people, 38%, chose immature female.
Okay, that's correct. So immature female, and we see 2 different types of oocytes here. These are our stage 1, our primary growth oocytes. And over a little closer, this bigger one with the vesicles in it, this is a stage 2. Okay, there's probably other stage 2s visible as well, maybe down there. But just know that any gonad that just contains stage 1 and stage 2 is immature. It has to have stage 3 or 4 to be considered mature. Okay, and practice question number 10. What did people choose?
65% chose ovotestis.
Yeah, that is a good choice here. So we can see that we're zoomed in at 400x probably. These are actually gonial cells, whether oogonia, probably spermatogonia, hard to tell. These are primary growth oocytes. We see in immature females, but we also see them in mature fish. And then we can see over here what looks to be spermatogenic tissue. So in that case, if you saw more of that and you saw that typical testicular architecture, we would call this an ovotestis. Very good. Okay, so that concludes the 3rd module here. I believe we might have some time for questions. Is that correct?
We have 2 minutes for questions before Ken start. So I will ask, just a minute, Pak Billy Theodorus Wage, silakan Pak. Pak Billy, silahkan tolong nyalakan mic-nya. Oke, I'll pass. Yang berikutnya, Ibu Hetty Hartati. Silakan Ibu Hetty Hartati.
Hello.
Yeah.
Yeah, thank you. So my question is, what about for the fish that on the postponing or resting, we may consider it as immature, right?
That is an excellent question. Some people have a classification scheme that draws on the thickness of the ovarian wall to indicate when a truly spent individual may still have been mature in the past. It's possible that our schema of just using the oocytes could miss individuals that are completely spent, but it is our experience that usually these individuals still retain some vitellogenic oocytes, at least in the tropics. Ken, what do you think about that?
I think that's correct, that we rarely find larger individuals that only have stage 1 or stage 2 oocytes. However, I think the other part of the question was a true resting phase, and I believe we did have some difficulty one time with Plectropoma species, a coral trout, where we're not convinced that we were able to identify size at maturity because accurate size at maturity, because we had so many large individuals that only had stage 1 oocytes, which is probably indicative of true resting. But we've also seen people say that a thick-walled ovary is indicative of prior spawning, and we've seen people say a thin-walled ovary is indicative of prior spawning. So there seems not to be a consensus about what to do about that ovarian wall and what that may indicate.
No. I think the best idea is just try to base your collections on times when you know the spawning season is occurring. That would be the best time to estimate L50.
And just to chime in too, Ross, I would say making sure that you have a high enough sample size that if you sample enough fishes, that you'll most likely see, even if some of them are in a resting phase, others will exhibit basically the stages of either the eggs or testes that will be able to match with the size structures.
Sure, absolutely.
Thank you. I think now is Ken's time. Go ahead, Ken.
Okay, thank you all very much. I'd like to close out by spending a few minutes explaining where to find the exam. We had some questions in the Q&A portion asking whether people could get access to the standard operating procedure that Eric and I have both mentioned. And I'd just like to be sure that everybody can find where that is, including, I know that Ross said earlier on that many people had gone to La Lima already and were finding their way to the exam. That's very impressive. But in case or in the event that some people have not been able to find out or to figure out how to log into La Lima, I'd like to go through that process now. So I'm going to go over to the site that you should have, whoops, sorry, I'm on the wrong one. I'm going to go over to the site. This is a fake email account that I created. And I'd like to show you what it should look like when you get an invitation to join La Lima. And La Lima is where you're going to find the resources and the exams. So basically, you should have a message that says, "Welcome to La Lima" from the postmaster at hawaii.edu. And in there, there's a link that says, "Accept this invitation." Just click on that and you're going to get a login screen. So I'm going to show you what I'm going to do. You type in your name, and the name would be as you want it to appear on your exam or on your certificate that you receive. So I like to go by Ken. I'm going to put in my first and last name. I'm going to create a password. And they're going to make me confirm that password. And then it just says activate your account and log in. And you should have received instructions on how to do this attached to an earlier email. So when you first log in, you don't see the histology training site. You have to go up here to this area marked sites and choose histology training. And then when you do that, go to organize favorites, and it says automatically add the new sites to your favorites bar, turn that to on, and then you close this out, and you need to reload to see your updated favorites site preferences. And now here you can see that there's a histology training tab at the top of the bar. Click on that. I'm not sure why it says that. Anyway, you can see you can have tests that you can take. I'll come back to this in just a little while, but the resources are what we're going to be after. Let me get myself out of the way. And here is the standard operating procedure. You can click on that and you'll be taken to a site where you can download the standard operating procedure currently in English. As soon as we have the full Bahasa Indonesia translation, it'll also be available in Bahasa Indonesia. These are files that we're going to use for the third and fourth sessions, so don't worry about those right now. But I will talk about later on which ones you need to access. But for now, if you want the standard operating procedure, it's right there. It provides every information on everything we've talked about today, and oftentimes a lot more detail on what we talked about today. When you want to take the exam, you just click on assignments, tests, and surveys, and you click begin when you're ready to go. Now, this is an exam made up of 30 questions. You can take the exam an unlimited number of times, but the 30 questions are randomly drawn from about 300 or more questions. So every time you take the exam, it's going to look a little bit different. So don't think that you're going to be able to go in and do the exam one time and record the answers and get a passing grade because you've got all the answers. Rather, it's going to look different every time. So our hope is that you'll do this as often as you need to get the passing grade, and that everybody gets the certificate, and that this whole test-taking procedure will be a learning experience. So when you're ready to begin, just click on begin. I'm going to get out of your way again. It provides some instructions in both English and Bahasa Indonesia. Whoops. And then you just click on begin. And what you'll see is that every question is basically like what you've been doing on the polls so far. There will be an image and you will be asked to identify it. Ooh, I'm under a little bit of pressure here. I'm going to call that an immature male. And then you just click next and you can go on. I'm not going to go through the whole exam right now. I just wanted to show you how you take the exam. So there'll be 30 questions. I'm going to press finish. I'm sure I want to finish. I know that I haven't answered all the questions. I'll continue that later. I truly want to finish. I really, really want to finish. And then I can go to the review. So because that was just one question answered, I'm going to go to another account that I have in La Lima, where I already did the exam. And here I can go and I act as if I've clicked the review, and here's what you'll find. For those of you who are answered in the certificate, here's the area where you get your certificate. And if you click on that, your certificate will pop up right away, available for you to complete. And again, I had a fake name. I just did this as test login. So it put my name as test login. So whatever you put on those in those spaces when you first create your account is how your name is going to appear. In the review process, if you scroll down, you can look at all the questions. You can see what you answered, and then there will be some feedback to each of those questions. So if you got the question wrong, we will explain to you what it truly was. Here's another one. This one could be done with words because all the oocytes were in stage 1. There was really nothing to point to. Here's one where the answer was mature male. And here's the feedback, but the bar is cut off a little bit, so you'll have to scroll down to see the whole answer. And it says, if you look at the place where the yellow arrow is, you can see the mature spermatozoa. So we're pointing to where the mature spermatozoa are. So hopefully, if you're having difficulty and didn't answer the question correctly, you can even learn from your mistakes here. And that is all I had to say about the exam and how to get into El Alima. So I hope you enjoy taking the exam, and I hope you enjoy the SOP. And thank you very much.
Thank you very much, Ken. Thank you, Eric, and thank you, Ross, for the very interesting training material presentation. Actually, I learned a lot too from the training. And ya, Bapak Ibu, terima kasih atas partisipasinya. Tadi itu saya baru tulis di chat alamat registrasi yang kemarin Bapak Ibu isi. Tolong diregistrasi ulang ya, karena kami ingin mendapatkan nomor WA Bapak Ibu. Beberapa peserta yang mendaftar mengisi emailnya typo, salah ketik. Jadi ketika kami kirim email, bouncing, cukup banyak juga yang typo. Jadi kita tidak bisa follow up, dan oleh karenanya kami butuh nomor telepon WA untuk bisa memfollow up. Mungkin itu untuk sesi hari ini, tanggal 18 Januari 2021 untuk modul 1. Berikutnya modul 1 yang kedua akan diberikan pada tanggal 21 Januari 2021 pada jam yang sama, jam 9 pagi waktu Indonesia bagian Barat. Terima kasih Bapak Ibu. Thank you very much for all the panelists, the presenter. Saya Eni Buhari mau pamit undur. Terima kasih.